The overall goal of this procedure is to develop manufacturing cell lines for biologics with clonality and high productivity. This is accomplished by first transfecting the host cell with DNA encoding the gene of interest. The next step of the procedure is to isolate high-producing clones by FACS, followed by documentation of colony formation by the CloneSelect imager. The final step is to screen and select highly productive clones. Ultimately, results can be obtained that show antibody production in continuous and fed batch cultures through reverse phase HPLC.
Visual demonstration of this method is critical, as the FACS sorting and TECAN-aided ELISA steps are difficult to learn. Because a complex setup is involved in these procedures, the following scientists from my laboratory will demonstrate these procedures. Liang Deng for transfection of the host cell, Feng Hong for FACS sorting, Jason Saunders for documentation of colony formation, and Russell Condon for TECAN sampling and ELISA.
To begin this procedure, 1 day before transfection, seed the host cells in a cell culture flask in 15 milliliters of growth medium. Incubate the cells in a 37 degrees Celsius, 7.5% carbon dioxide incubator. On the following day, prepare the transfection complexes. First, dilute 8 micrograms DNA in 800 microliters of growth medium without serum in a sterile microcentrifuge tube. Mix gently. Next, dilute 40 microliters lipofectamine in 800 microliters of growth medium without serum in a polystyrene tube. Add the diluted DNA